I'm Jay Horton. I'm a gastroenterologist at UT Southwestern Medical Center in Dallas, and I'm here with my co-chair, Alice Chang, who's associate professor in the Department of Endocrinology and Metabolism at the University of Toronto. And I'd like to welcome everyone to this industry satellite symposium. And everyone can see the slides now. I'd like to, as I said, welcome everyone. This is being sponsored by Pfizer. And as shown in this slide, it's been 35 years since Brown and Goldstein were awarded the Nobel Prize in Medicine for their discoveries, which, in quote, was for the regulation of cholesterol metabolism. And this occurred in 1985. I believe this was the 13th Nobel Prize that's been awarded for studies related to cholesterol. And shortly after the Nobel Prize was awarded, actually in 1987, Merck capitalized on many of their discoveries and ultimately developed lovastatin, which of course was the first statin that was approved for use in the United States for the treatment of hypercholesterolemia. The goal of the symposium is to highlight how subsequent studies in the field have led to the development of several new exciting therapeutic options to both prevent and treat cardiovascular disease. So I'll now turn the program over for further introductions to my co-chair, Dr. Alice Chang.
Great. Thank you very much, Professor Horton, and welcome to everyone to this symposium tonight. And it's my pleasure to share the stage with these giants in their respective fields. And it's my pleasure to also introduce, again, since you've already heard from both of them tonight already, Professor Robert Hegley as well as Professor Bernie Zinman, both of whom are friends, are colleagues, as well as mentors. And in the case of Dr. Hegley, he is an endocrinologist as well as a lipidologist from Western University. And in the case of Dr. Zinman, he is also known as Uncle Zinman, or Uncle Bernie is what I like to call him as well. So he's certainly been a mentor of mine for many years to come. So I look forward to hearing from both of them. And in terms of disclosure for tonight's symposium, Pfizer provided financial support for this scientific exchange symposium. Presentations were developed by each faculty member independently of the sponsor, and each faculty member had full editorial control over the content of the presentation. And tonight, in terms of the lectures that you're going to hear from each of these giants in their fields, the titles are listed for you here. I won't read them out to you, but they're going to be brief presentations because we wanted to make sure that there was lots of time for discussion. So I encourage all of you to please type in questions in the chat box and we'll be sure to address them. So without further ado, let's kick off the videos so that we can watch these lectures.
Hello, I'm Jay Horton, and I'm a professor of internal medicine and molecular genetics at UT Southwestern Medical Center in Dallas. And I've been asked to discuss the evolving focus in the Department of Molecular Genetics that has gone from the discovery of the LDL receptor to PCSK9 to work in fatty liver disease. Here are my disclosures. So it's been 35 years since Brown and Goldstein were awarded the Nobel Prize in Medicine for their discovery and characterization of the LDL receptor. I've been asked to provide a brief summary of the work done in their departments since they were awarded the prize. So I'll start with a classic slide that illustrates the thrust of their research efforts both before and after being awarded the prize. Their primary goal has been to determine how cholesterol homeostasis is maintained within the cell and the whole organism. Their early work focused on two primary paths for cholesterol accumulation: endogenous synthesis and extracellular uptake, which of course is mediated by the LDL receptor. Once the LDL receptor was discovered, they demonstrated that cholesterol also regulates LDL receptor expression in mRNA levels. So they then set out to identify the transcription factors responsible for this sterol-mediated regulation. This led to the biochemical purification of SREBPs, or sterol regulatory element binding proteins. These transcription factors were unexpectedly found to be membrane bound, and they had to be cleaved in order to become active. Over the past 25 years, Brown and Goldstein have identified and characterized the key proteins that carry out this complex event. Central to cholesterol regulation is the SCAP protein, which binds both cholesterol and SREBPs and serves as an escort protein that transports SREBPs to the Golgi, where they are cleaved and subsequently activated. An additional focus in the department has been on the characterization of genes that are regulated by SREBPs. There are 2 SRBP isoforms, and work done in the department and by others have demonstrated that SRBP2 not only regulates the LDL receptor, but it also regulates all enzymes involved in cholesterol biosynthesis. It was through array studies using livers of SRBP2 transgenic mice that we identified NEST, which turned out to be PCSK9, which was SRBP2 regulated. And ultimately, this protein was shown to post-transcriptionally regulate LDL receptor protein levels. The SRBP1 isoform was found to regulate all enzymes involved in fatty acid synthesis and the first enzyme in triglyceride synthesis. And it was the overexpression of SRBP1 that resulted in fatty livers, and it was this finding that ultimately served as the impetus for further studies that defined the contribution of de novo lipogenesis in the development of non-alcoholic fatty liver disease. Additional members in the department, Drs. Hobbs and Cohen, have used genetics to identify the function of new genes in lipid metabolism. They relied heavily on the Dallas Heart Study participants who agreed to have multiple phenotypic parameters measured, including plasma lipids, DNA, and NMR measurements of their liver fat. Using the DNA provided, Hobbs and Cohen identified loss of function mutations in PCSK9 that were associated with low blood LDL cholesterol levels, and subsequently they showed in a different large population that those harboring the loss of function mutations had an 88% reduction in cardiovascular events. They've also used GWAS studies from the Dallas Heart Study to identify variation in gene sequences that are associated with high liver triglycerides. A variation in PNPLA3 was identified that was present in more than 20% of the population. This is a lipase, and others have subsequently showed that this polymorphism in PNPLA3 is not only associated with hepatic steatosis, but also with NASH, cirrhosis, and the development of hepatocellular carcinoma. Ironically, PNPLA3 expression is also regulated by SRBP1, which brings us back to the same fundamental pathway. So in summary, the research carried out in the department has continued to focus on defining how cholesterol regulates whole body cholesterol homeostasis at the molecular level. The characterization of genes regulated by SRBPs have led to the characterization of PCSK9 and to defining the role of SRBP-regulated genes in the development of non-alcoholic fatty liver disease. Both pathways are now being exploited to treat hypercholesterolemia and non-alcoholic fatty liver disease, respectively. Thank you for your attention. I'll be happy to address any questions.
Hi, good evening. I'm Rob Hegley from the University of Western Ontario. In the next few minutes, I'm going to be talking to you about new drug targets for cardiovascular disease. These are my disclosures for this evening. The drug targets, PCSK9, ApoC3, ApoA or LPA, ANGPTL3, have all been shown through genetic research primarily to be risk factors for atherosclerosis, and some of them are actually acting through lipid levels. And these then, when we know that the levels of these targets are high, if we can knock them down, we can reduce risk. And so the strategies to knock down are through RNA interference in some way or monoclonal antibodies. Now, just to remind you, there are 2 ways of targeting RNA. You can target it in the cytoplasm using a short interfering RNA, or you can target it closer to the nucleus using an ASO or a allele-specific oligonucleotide. So the first drug target is PCSK9, very, very well known to you. There are already 2 drugs in the market around the world. These target PCSK9 that is circulating using a monoclonal antibody approach. There is now a new way to target PCSK9, and that's using the RNA interference approach, and that's within the cell, and that's this agent inclisiran. So inclisiran is a short interfering RNA. In this particular study, it was given 4 times over an 18-month period. It reduced LDL cholesterol by 50%, sustained reductions, and in fact, it's very, very likely that this drug will be approved quite soon in North America and around the world. The second target, ANGPTL3, angiopoietin-like 3 protein. Through genetic studies, we know that individuals who are deficient in this particular protein have low levels of lipid, so that both triglycerides and LDL, so that by targeting it, either through antibodies or through RNA, we try to recapitulate this beneficial human phenotype. So there is a drug, a monoclonal antibody, evinacumab, which amazingly lowers LDL in patients with homozygous familial hypercholesterolemia. Another drug that targets RNA, buprenorphine, similarly lowers triglycerides and lowers LDL. It basically causes improvement in multiple lipid fractions. So these are both now primed to be tested in actual cardiovascular risk reduction studies. The third target you may have heard of, Lp(a) or apo(a). So this is an LDL-like particle that raises risk, also liver secreted. It can be now targeted specifically through RNA targeting. So this is pelacarsen, and again showing a dose-related response. And these people, for the first time, can get extremely amazing reductions in Lp(a). So this is now ready for prime time to be tested in cardiovascular outcome studies. And then finally, ApoC3, a drug or a target that is associated with high levels of triglyceride. So a number of monoclonal antibodies or a number of RNA interference drugs now that lower ApoC3 and lower triglycerides by 80 to 90%. So the four targets, there are various evidence, both from genetic evidence, other kinds of scientific evidence. The names of the drugs, the targets, they're all shown on this slide. The effects on the lipid profile, some of them have already been shown to have effects on cardiovascular disease. So the ideal target to be targeted either by a monoclonal antibody or by RNA interference are liver secreted, they raise risk, so then when you knock them down, you can lower risk. There's fantastic evidence for the four targets that I've talked about this evening. These actually then work by reducing the end product, the lipid level, LDL, triglyceride, Lp(a). If the triglycerides are severely elevated, there's risk for pancreatitis, and so that's another clinical endpoint that can be helped. So we are now on the verge of having ready for prime time RNA interference drugs. For the future, we can talk about gene editing, but that's a topic for another day. Thanks for your attention.
Hello, I'm Bernie Zinman, and I'm delighted to participate in this symposium. I am a senior scientist at the Luna-Phil-Tannenbaum Research Institute and professor of medicine at the University of Toronto. My topic is evolving science in cardiovascular risk reduction in type 2 diabetes. My disclosures are shown on this slide, and I serve as a consultant and receive honoraria from companies that are involved in diabetes research. Now, life expectancy is reduced in patients with diabetes. And this slide shows that for a 60-year-old individual, having diabetes and cardiovascular disease means a 12-year reduction in life expectancy. Quite dramatic. Now, heart failure is often not recognized as a common complication of cardiovascular complication of diabetes. And what we see here is that for individuals with type 2 diabetes who are using insulin, so that means that they're pretty far advanced in the course of diabetes, there is much more heart failure occurring than myocardial infarction or stroke, and heart failure can have very devastating consequences. So if you were thinking about a diabetes therapy and you were having a wish list, this is what it could look like. You want effective glucose lowering, low risk of hypoglycemia, no weight gain, complementary mechanisms of action with other diabetes therapies, safety, well tolerated, and I've shown here in bold some of the added value which would really be a bonus if a diabetes therapy had a beneficial effect on lipids, had a beneficial effect on beta cell function, and actually changed cardiovascular outcomes. That the molecule itself was special. And indeed, the concern by the FDA at the time was with respect to safety, because there were diabetes therapies like thiazolidinediones that were associated with increase in heart failure, with a concern that there may be an increase in cardiovascular safety. And here you see a whole slew of trials involving hundreds of thousands of patients of the GLP-1 receptor agonists, the SGLT2 inhibitors, and the DPP-4 inhibitors. I'm going to focus principally on the GLP-1 receptor agonists and the SGLT2 inhibitors. So the world changed in 2015 at the EASD in Stockholm because this was the first trial to report not only safety, but of benefit. And here you see the effect on the primary outcome of empagliflozin in the EMPA-REG outcome trial, a 14% reduction in MACE, a reduction in hospitalization for heart failure by 35%, a reduction in cardiovascular death by 38%, and a beneficial effect on renal function over the duration of the trial. So Darren McGuire did a nice meta-analysis looking at MACE, and what you can see here is a very positive, beneficial effect in the pooled estimate. Most of the trials, except for Virtus, show a shift to the left with respect to MACE. Heart failure hospitalization, even a more robust benefit shown in all the trials. Let's look at MACE with respect to GLP-1 receptor agonists, and you see a benefit that is not consistent, but demonstrated with most of the trials. As far as heart failure hospitalization, there is no consistent benefit. So these trials, as well as many others, provided the evidence for the American Diabetes Association and other societies to develop guidelines. And here we see that, in fact, the American Diabetes Association in 2021 recommends that for people with risk factors or established atherosclerotic cardiovascular disease, CKD or heart failure, that one goes along the path of a GLP-1 receptor agonist or an SGLT2 inhibitor. And that is for individuals with atherosclerotic cardiovascular disease or risk factors. However, if heart failure is the primary concern, then an SGLT2 inhibitor is preferred. And for CKD, similarly, the data is stronger for an SGLT2 inhibitor. So what I've tried to do briefly is provide you with the evidence that has informed not only clinical practice guidelines, but our day-to-day management of patients with type 2 diabetes, and has resulted in an improved outcome, particularly as it relates to cardiovascular disease. Thank you so much for your attention.
Thank you very much, gentlemen, for those fantastic presentations, sort of a whirlwind of lots of research and information packed into short, concise presentations. So thank you very much for that. I certainly invite the audience to ask any questions that may have come up as you were listening and enter that into the Q&A. And I'm going to actually start with Uncle Bernie. So I have a question for you, sir, about the realization of all the great stuff we have. And this was alluded to actually even in the first session, but I mean, you clearly showed we have tremendous data. Millions and millions of dollars have been spent. More importantly, thousands and thousands of patients have volunteered their time to be part of these trials. But yet our use of these outcome reducing therapies are still sitting at about 22% in that recent analysis looking at multiple different countries. So why? Why do you think that is, and what can we do to try to fix this?
Yeah, clinical inertia is a huge problem, and obviously symposium like today and continued education is critical. But just to give you another example, when the DCCT results were presented in 1993, that intensive therapy with 4 injections or a pump was clearly the treatment of choice for type 1 diabetes. It took years until that became adopted universally. And so the SGLT2 inhibitors, the GLP-1 receptor agonists are being used with greater frequency, but not enough yet. And so it's very frustrating for us who are involved in the research to see the slow translation of critical research findings. So we need to just keep plugging away at this, but I think it's classic clinical inertia. People don't like changes.
And I think the multidisciplinary learning is going to be critical, right? Because certainly these therapies don't belong to diabetes, certainly not anymore. They've gone beyond those walls for sure. I'm going to turn to my co-chair, Jay. I understand you had some questions for Dr. Hegley, for Rob. I'm going to let you fire away with that, and I hope they're tough ones too. Go ahead.
Well, it's been a while since I've had a chat with him, so we can catch up. But please, others, feel free to type in questions for us. But start with, we'll start with the emergence of the PCSK9 antibodies, and now the recent approval of the ANGT-PTL3 antibody as well. They're both very effective at lowering LDL, and in the second case, in lowering VLDL as well. But what do you think needs to happen for these agents to actually get routinely used, not only on top of statins, but particularly in statin intolerant patients, which I know there's a lot of controversy around, but the bottom line is, you know, if the patient isn't going to take the statin, they are going to take the statin. And so what are your ideas about how these will actually become more commonly used in these conditions?
Yeah, so thanks, Jay, and it's nice to see you again. So I think the big thing is accessibility. Like just even off the top. So I mean, if Kiso was approved, you know, a couple of weeks ago, the ANGPTL3 monoclonal, but the cost I've heard is just enormous. Now it's for a very, very limited indication, homozygous FH, and it's great. It is a life-changing treatment for those patients. But right now, you know, it's the cost. And then the same thing when the PCSK9 monoclonal antibodies came out. I mean, I've got 250 patients on them. They're, you know, mainly heterozygous FH. They are life-changing treatments. Patients love them, actually. I thought there would be a big issue with you know, with getting people to wrap their minds around it, but in fact, you know, it's been very, very well accepted, and you know, no issues with tolerability, but you know, the main thing actually is just a practical point of access and coverage. And then you know, experience, and then you know, getting our colleagues more experience with prescribing them. So I think it boils down to maybe more practical issues of implementation.
So while I have you, I'll just ask another implementation question, and that's the Lp(a) therapy. That looks very impressive as well, very potent. But how will that be used? Do you think there's going to be an absolute target? Or, as you pointed out, many of the high-risk people that you see have astronomical levels of Lp(a), where a 50% reduction, albeit good, still keeps them astronomically high.
Yeah.
And so how do you anticipate this will ultimately be used?
Yeah. So I think I mean, the clinical trial is being, I mean, there are actually a couple now that are ongoing. Actually, there's two agents, Amgen and Novartis. And so it looks, the entry criteria are based on high levels. So I think it's going to be a matter of, like, the higher you are, the bigger the benefit. And so then that's going to be necessarily a smaller proportion of the population. And so, you know, Lp(a) has this very skewed distribution in the population. Most of us are sort of, you know, fortunately have a low level. So, you know, we wouldn't need that drug. There would be other forms of residual risk to try to target. So I think, I actually believe that those studies are going to be positive, but then I think necessarily it's going to be a restricted market for who's going to use them.
Now, I have a question for Jay, actually. And as a gastroenterologist, you're now facing an audience full of endocrinologists and cardiologists and nephrologists, and have an opportunity to give us messages around fatty liver, because we certainly, all of us, are seeing patients with fatty liver and probably missing. Most of them are not recognizing it or not doing the right things about it. So what do you want to tell us so that we can start doing things better?
That actually brings me to Dr. Zinman, because I was hoping to see NAFL on his slide. He had a very nice slide of additional desirable characteristics of anti-hyperglycemic medications, and I think NAFL should be on there as well. And certainly a couple of them that you discussed show, you know, my personal opinion is one of them is likely to be the first one that actually is approved for the treatment of fatty liver disease. But but that's personal opinion, not based on any real data yet. But nevertheless, you're right. I mean, the 80+% of the patients you see in your diabetes clinic is going to have a fatty liver. And the question that we all struggle with is who to treat, when, and how. Currently, as most know, the entry criteria for the fatty liver trials are very strict and require a certain type of disease, which the majority of people don't have, fortunately. But it is very restrictive, and then showing improvement becomes even more difficult. But I think that there are a number of drugs under study. Many of them are to reduce lipogenesis, as I discussed in my talk. But I have the same question with fatty liver as I did with Rob with Lp(a), whereas a 50% reduction in these trials is very good. But if you're starting at 60% liver fat, is getting to 30% liver fat, does that really matter? And we don't know the answer to that question. And does the absolute liver fat matter? You have inflammation, and of course, the cirrhosis is what most people are interested in. But I'd also be interested in Dr. Zinman's ideas, since I personally think that the most likely course of a successful drug is going to come through some of the drugs that he discussed.
Yeah, I think you're absolutely right. I think that there are two ways of approaching the fatty liver issue. The NAFL and NASH is in the context of the new therapies for diabetes have robust effects on weight, particularly if you look at the GLP-1, GIP dual agonist, tirzepatide as an example. You're talking about 12 kilogram weight loss, more than 10, in some patients more than 10%. I think that's going to have a huge impact on the hepatic biology. In addition, as you point out, there may be some diabetes therapies that have a direct effect on hepatic fat. And you're absolutely right. We're going to have to modify that slide, as that's another added value that we should be looking for. So not only do we have diabetes therapies that fall under nephrologists, cardiologists, now, of course, it will be hepatologists as well.
So the question from the audience here, and I'm going to direct this to Rob, asking about lowering LDL levels to below 0.5 and keeping it there for a longer period of time. Do we have data about safety for doing that?
Yeah. So, I mean, we're starting to get data. So I think, like the extreme would be the person who has zero LDL. Okay. So we know that that's not good, A beta lipoproteinemia, but some LDL, like 0.3, 0.5, I mean, that's, that's, even in infancy, that's quite a common level. And, infants are developing okay. So now with PCSK9 inhibitors, I'm routinely seeing patients with levels that low, and I'm just letting them go. From the subgroups from the clinical trial show that it's safe. Neurocognitive assessments show that it's safe. So, I mean, over the short term, over the window of where you're trying to prevent an MI, say, a 3 to 5 year period. Now, we don't know over 20 or 30 years, but we're so I think the more experience we gain, the safer we feel about it, and it's so the whole distribution is shifting.
Okay.
Do we have time for maybe a couple from the audience? And one of the first ones that appeared was a very basic question: why use metformin first?
Yeah.
It doesn't have the added value from your list. Yes, it's safe and cheap, but not effective in reducing some of the secondary things you discussed. So, Dr. Zinman.
There actually is a movement to move away from metformin, and the European Cardiology Society actually recommends that it not be the first. And our co-chair, Alice, will be debating metformin in the next section of this presentation, so whether it should be used or not. It's hard to displace mother metformin. We all love it because it's safe and it results in weight loss, no hypoglycemia, it's effective. So wait to hear that debate.
So I guess there's one more. I'm not quite sure I understand relevance to perirenal fat, which exacerbates primary hypertension. I'll let Alice or Dr. Zinman maybe try to address that.
I'm going to let Alice address it.
All right.
I was going to let Uncle Bernie address that one. So I don't know that I can comment on that one.
Well, actually, people have looked at fat. We always think of fat in the liver and in the viscera, but people have looked at pericardial fat, and there may, that kind of fat that is either in the organ or around the organ is certainly not the kind of fat you want. And the subcutaneous fat seems to be innocuous, I guess, is a term we could use, but it's they're different kinds of fat, and it's very metabolically active.
So I want to take this opportunity to thank all three of you for a fantastic presentation, and thanks the audience for their questions, for an engaging panel discussion, and of course thank the sponsor, thank Pfizer for the opportunity to be part of this symposium. And I encourage everyone to stay tuned for the next section of the At the Limits conference. Thank you.